them.
Oh yeah, you're kind of like me. There's a little cinnamon in here.
Smell it.
Can you?
Yes.
Can you smell the cinnamon?
I can.
Okay, good. Now we just make at home. We make this regular coffee, but we found if you put some powdered cinnamon in the grindings, it's flavored.
I'll have to try that.
Oh, you'll have to try it, yeah. Well, because we do a combo. We have coffee beans, but then we also buy ground up stuff, and so then we'll put a part in the ground up can. Then we'll put cinnamon and mix it up so there's got some in there. And the beans don't have any cinnamon in it, but sometimes they have hazelnut flavor or whatever. But yeah, I'm just spoiled now. I can't. And then this has a little half and half in it. Are you a half and half person?
Not really.
Okay, you just take it straight.
Pretty much.
Yeah. Well, that reminds me, I used to live, when I was, part of my life, I used to live in this basement apartment when I was an undergrad. And the people that owned the house I rented from, they lived on the first floor. And every time they made popcorn, the smell would come down, make me want popcorn. So then I had to make popcorn every time they made popcorn. Because it was like, oh man, popcorn does sound good. So it's kind of like that. Okay, I guess I'm going to start. I can't remember if I said I'm going to start at 4:35 because the 10 minutes doesn't get everybody, but I'll just start with some general things. And if people mosey in, that's great. I'm going to always try to remember to do the microphone so everybody can hear well. And one thing about today, I don't know if you noticed, we have a dog here, but it's a dog that you don't pet. It's a dog in training to be a service dog. And the dog was also in one of my other classes, and we learned that they're supposed to be trained and center on who they're with, but they're not supposed to interact with anybody else. So that's why if you see the dog there, it says ICAN, it stands for Indiana Canine Assistance Network. So it'll be here, I don't know how often, but anyway, so I'm the one that has to train myself not to pet animals that are doing service in our training, because I'm the worst violator. Oh, let me touch your dog and let me pet your dog. So just as a thing, if you ever see a dog that's probably being trained for service like this one is, then the key is you ignore it, because it's supposed to be in training such that it gives all its attention to the person it's with. And you can kind of see why, then if we pet it, then it'll be distracted, and then when it gets out in the real working world, it might want to be petted by other people. See what? So it's a great strategy of, leave the dog alone, let it focus. And then the other thing is, when dogs are in training, this is a little bit animal behavior, not 100% get the final diploma. Sometimes they fail. And I remember meeting a dog one time that if you sneezed, it would go to the Kleenex box and bring you a Kleenex. And then when you dropped the Kleenex on the floor after you used it, it would pick it up and take it to the garbage can. It was that good. But it failed one of its last tests. And here's how it failed. Whenever it was out in the real world, it was too distracted by other dogs. It would almost like want to go play with the other dogs. And of course, if the dog is a service dog, it's supposed to be assisting you and not be distracted. But what a dog to adopt. So this one person that I knew adopted the dog because it couldn't be used for service, because it had failed. The only thing it failed was it got excited at other dogs. So, I mean, it was trained to do all open drawers. So interesting. So anyway, how many watched the Body Worlds video? How many have seen it? Anybody? Did you see the whole thing, or?
Oh, wait. I thought you were talking about the videos for the readings. this week.
Oh, I see. No, the body worlds, I think I put it up in Brightspace. Can't remember. Did anybody see it? Well, you saw it, well, the reason I'm asking is not so much, because it's totally optional. I just want to make sure, sometimes what I see on my computer, it's not the student view. So it was accessible, is basically the bottom line. Okay, excellent. So before we continue on, if you try to get to my website, I messed up this morning. I was reading emails, and my website is overwritten by another file, but tonight I'll fix it. But you could get to this thing. I guess this is, look at it, it's called rawaugh30.com. Remember, there's 3 websites, but I messed up this morning. And when I FTP to these servers, I don't really have anything at Purdue that can do it. I've got everything set up on my home computers. So anyway, I'm going to go to our website, but I'm not doing the regular route. But before I do that, anybody have any questions? Burning questions about anything? Nobody? Not a one question? Okay. Okay, so let's go here, because I want to talk about some of this stuff, and then let's see. I think there's a few people in here that want to do honors, so there's the honors syllabus. There are people that do courses in honors mode. I did that, if that affects you. I'm starting to fill up this page called dissection guides. And the TAs are helping me too. The TAs, and some of them are in the room now, but if they weren't at that one meeting we had last week. So if you're a TA in this class, try to find good websites that are either videos or text that talk about dissecting any domestic animal. Dog, cat, horse, pig, rats. Rats are, well, I guess they're domesticated in the sense the lab rat is domesticated. And then I'll put it here. I guess let me click on it while I'm doing this. So what I did is, it's under construction, but before we start really dissecting, tomorrow we have lab, but we're doing a dry lab, by the way, so you don't have to have a lab coat or safety glasses. We're going to be talking about how we conduct the labs, and we'll start out with some maybe microscopy. But my goal here is to have this as a resource for you, although we're not going to be dissecting cattle. We're going to do rats, pigs, and pregnant cats. But the techniques are often very informative, and some of these are videos, and I won't click on them. But at the bottom here, I did put some links to the body worlds. Remember the horse I showed you? I showed you the horse last week, didn't I? Yeah. Because that plastination is amazing. It's got the same odor as this bottle. Nothing. There's no odor to this process. And then I know Gunther started doing it with more animals. He works at a med school, so he's doing human anatomy, of course, was the primary, but they've been, I showed you the horse that they did, and then they're doing other animals, but I cannot find any video of the animals. But I did load up in the Brightspace. If you go to Kaltura Media Gallery, I think it's called, then you can see the process. It's very neat. It's hundreds of hours, terribly expensive, and I'm not even sure if some of the animals that has done have filtered into the vet school. And I should, I could text some vet students and see, are you guys doing anything with plastination? But it's really quite a process, and it's neat. Okay, I'm going to get rid of that. And so then, remember, I'm going to do the reverse time. There's last week, and then here's this week. And what I want to say is, the syllabus says we have 4 exams. They're all listed there when they're due. You're going to get them probably about 5 days before the due date. Then you can upload them into Brightspace. There'll be a space there for it. You can upload a better version if you want to do another version and get your similarity score down. The syllabus says you should have 20 or less for a similarity score. It's impossible. Sometimes people get 1 or 2 or 0, but there's certain things you say, like we're gonna do some kidney stuff, that, there's no other way to say it, but the functional unit of the kidney is the nephron, okay? But here's the kicker, whenever I start talking about things in this class, I expect that stuff to show up in lab reports and the test. So that's gonna be a little different. You're gonna make a lab report on kidneys. But whatever I say today should be reflected in that lab report. It's not like labs are separate from lecture. No, it's all together. So like when you do a test, if you did something in lab that can be brought into the test paragraph, then you should do that. Why? Because it all should be one bundle. It shouldn't be separate. Okay, it shouldn't be like, let's say you do the pregnant cat and talk about the uterus and the fetuses and all that. There might be a test question on something similar that we talked about in lecture or have read. You can incorporate it all. You could, be talking about the ovaries and say, yeah, in the pregnant cat, there's 2 ovaries, and I counted the number of eggs released by those ovaries. Although it might be a month ago, we're going to show you how to count the number of eggs that were released by the ovary a month ago. And so when we do lab tomorrow, it's going to be dry, but I'm going to make sure you get into the mode of everything you touch, you take a photo of, animal tissue, you know what I'm talking about. You take a photo of, you weigh it, you measure it, you count it, whatever. It's not going to be one of these things where you dissect the animal and then you take a few pictures or you write up a lab report and no pictures. So pictures are good, videos are good, because maybe you'll do something with the pregnant cat and you'll never do it again. But then you have a video, you'll be working in pairs, and I think what we'll do is every week you have a different partner, because that's better than having the same partner. The same partner, maybe you'll learn something the first week, but the second week, nothing, you'll learn a few more things. But think of it as you have to switch partners every week. That's the best way to do this. But tissue is precious, and then every week that happens in lab, because I'm trying to orientate you to the lab, you won't make a lab report for that week. Lab reports are going to be on structures, like say kidneys. And it might take you a fair number of weeks to gather up enough about kidneys. And then in some labs, you'll be gathering things about the lungs. So you'll have multiple streams of information that then at some time down the line, you can do a lab report, and we'll talk about that in there. Anybody have any questions on that? You're going to come, it's in 10:45, it's just that way, and we'll see everybody then. Okay, so then with this reading, like last week I didn't say anything for lab, but here it is, look at for lab. Here's a good question, go ahead, make sure I hear it.
I was just going to ask what the dress code for lab is, like long pants, stuff like that.
Clothing is required. No. Now, yeah, that's a very good question, and it's a little bit of a dilemma because the room is used more for lectures than anything. And I'm struggling with this myself because this class usually just had 4 lectures and no lab, and then last spring it switched to 3 hours of lecture and 2 hours of lab, for 1 credit. And I didn't, it was somebody else. But I know there's a lot of, in this room, this is a long answer to your question, it's an orphan room to me. It means no one's in charge. And that's a little scary when you do things because things might disappear. So I've already got planned to do, some of my most delicate stuff I'm going to be having in my office, and I'm going to bring it down with a cart every day. Because if I put it in drawers, by the time, there's hundreds of people that go through that room in a week. I mean, thousands. I can easily see things walking away. It's not like a dedicated room where people have limited access. So, and then, if it's a true chemical lab, you can't drink coffee in the lab. And you're supposed to have closed-toed shoes and all that, no shorts. But this is my outfit for the lab tomorrow. I'm gonna have coffee. I'm gonna have shorts on. That's my dress code all year round. So it's not like a true chemical lab, let's put it that way. Like that for chemistry, right? If you guys did chemistry, you always had to have safety glasses, lab coat on maybe every time. But we really only need lab coats for when we're doing stuff. And I have some plastic aprons that will basically, you know, I don't want anybody to buy a lab coat when we're not going to use it every time, you know. And if you're careful, that's the other thing is I'm going to show you how to do these things with finesse. I'm going to show you how never to use force with a scalpel. I'm going to show you how to prop animals up so you can dissect them. But everything's going to be done with finesse. That means slow and easy and well thought out. That's why I'm combining, got some of those videos right there. But you're going to get trained how to do it, not do it fast. Too many people come and do something. And the other, see, so it's like, say you get a rat, pregnant rat, by the way. The worst thing you can do is dissect it, you know, and look at it and say, okay, that's fine. You're going to be getting parts out of the rat, but other people are going to get other parts out of the rat. So like some might be working on kidneys, some might be working on lungs, somebody might be working on the pregnant uterus. And it's going to take a while to do the pregnant uterus because you're going to weigh it, you're going to identify the ovaries, count how many eggs left each ovary, keep track of it left and right, keep track of the fetuses left and right, see if there's 5 ovulations on this side, do we have 5 fetuses? That's 100% embryonic survival. On the right, if there's 7 ovulations and only 3 fetuses, you had a lot of embryonic loss. Sometimes when the loss occurs after ossification, there'll be a little bump of evidence of a fetus. But if it's early loss, there's no evidence. So hopefully we're going to run into things where some fetuses aren't viable, and it'll show you that that was a fetus, but it's reabsorbing. It's called reabsorption. And the rule is early reabsorption can be complete because there's no bones to reabsorb. Later, if bones become hardened with calcium, then it's impossible to really absorb that in the length of a pregnancy. So we'll be teaching all those little nuances and not rushing, okay? Because then you can get, let's say you do the kidneys of a rat one week, maybe you'll get the kidneys of a pig the next week, fetal pigs. I'm not sure if they're fetal or just born, I'm not sure. They're the same, right? 2 days before birth is the same as 2 days after birth. I don't know how they do this. They're all fixed in a safe fixative, not formalin. How many have dissected an animal with formalin? Does it smell? Yeah, and it's not a good smell. This fixative has some smell, but it's not, I guess I'll say toxic like formalin. Formalin is kind of toxic. So you'll see how it goes over time. And then there'll be other tissue that I might bring from my collection. Like one time I'll bring a human uterus. Take it out of storage and show it to you. Or horse kidney. Most people haven't seen these things unless you've done a necropsy of these animals. And it's hard to get a hold of horse kidneys. How do you get a hold of a horse kidney? Unless you're someplace where they're necropsy. I used to visit a place that butchered horses for meat and collected a lot of parts. What was interesting is some of the horses were pregnant. Oh man, that was fantastic to dissect a pregnant horse uterus that had a foal inside. And we spent a lot of time dissecting the foal. It was like forever. So, you know, we're slowly going to work in the lab because there's not much there. Not much in the lab. And I don't want to bring a bunch of stuff and then leave it, and the next week my beautiful balances are gone, or, you know, things like that. So anyway, if you can, and it doesn't really matter, but if you can, try to watch those videos before you come to lab tomorrow. They're about anatomical planes and terms of orientation. And we'll be going through this in lab, so if you're busy, I understand it because it's 24 hours away. And these are videos I made. And the other thing I like about this is when I'm working with you, I might get some ideas of videos to make. Okay? And some of you this summer, where's Kale at? Is Kale, there he is. He's my hero. He did some things this summer that were fantastic, and he's got pictures, and he kept on sending me pictures. Remember I sent that email out and said, if you happen to be someplace where animals are dying, you know, not because you're killing them, but there's nothing better than a fresh carcass to dissect. And Kale's got some pictures. We'll be sharing them with some people. Yeah, anyway. So if you have your own pictures, even if you're working someplace this semester, and it's something about anatomy and physiology, take some pictures and put them in your lab report. Because these are going to be lab reports that you could show somebody else and show them the sections that we're going to talk about tomorrow. I don't want to steal that thunder. But in summary, last week we talked about blood, and you know 7% of the body weight is blood weight. There's 3 types of blood cells. And I think at the end of the semester somebody asked me, what did I mean by the term of packed cell volume, PCV, and that's really the percent of technically red blood cells in a sample, and by convention we're going to say 45%. So a normal packed cell volume equals 45%, and the packed cell volume is also called a hematocrit. A very common parameter measured at every vet clinic in the world. You know, when you take your animals in, they take a blood sample. And I probably should show you, every time my dogs go into the vet clinic, I want an email sent to me when the blood panel comes back. And you should do that too for your animals, because you paid for it, right? If your animals get radiographed, you get a copy of the radiographs too, you should. And you know, it used to be radiographs were like x-rays, and they had a lot of chemicals, they were processed, and they weren't available to anybody except who had the physical copy. Now the x-ray machines are digital. You can send radiographs to all parts of the world if you want to show somebody. So that's kind of neat. So you're going to read these things. I promote the note-taking topic. I'll show you maybe tomorrow in lab the link where I do what's called the atone. I think I've listed it someplace, where at the top of the page you put a topic, and then everything you learn in this class, whether it's in the lab or lecture, you write something down in a bulleted statement about that topic. And then in a 3-ring binder, I told you this, you keep them alphabetically. And then when you go to another physiology class, you don't have to think about, what did I learn about oxytocin? You go to oxytocin in that binder, and then you take that page out and take it to your next class that talks about oxytocin. Very powerful to accumulate things. Because I'm assuming a lot of people will have biology, physiology, anatomy in their future, right? I mean, if you're all going to be accountants, I wouldn't really talk that much about this. But oxytocin is a hormone. We talked about the 2 main types of hormones last time, right, didn't we? Steroids and protein, if I remember right. So remember, everything in lecture and everything in lab can be on every test or any test and in lab reports. Think of it as all one. So let's go back up here. The reason I'm doing kidney after blood is the kidney is an important organ, and it's going to make urine. So then I'm going to show you some structures that then in lab we could look at. It's kind of like old-fashioned to use a microscope now when all the histology is on the internet. I mean, why look into an eyepiece when you can get it up on the screen? So we'll go through some of these hormones and renal function. Sometimes when I don't want you to read the whole thing, I'll say stop reading blah, 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 and I might show you this if I can pop it up. You stop reading after you see the author's names, they're at the bottom. And then you got these videos, since I just put those up, I didn't have them up last week, you know, we'll talk about those in lab. But let me just do the anatomy of the kidney, okay? And so, and I don't have, so
Right. Did you download Grant?
I'm not logged into it, so I got
Come
these
on, man.
ad things
Everyone's
here.
using it.
All right, let's talk about kidney anatomy and answer
Okay.
the questions. What are the functions, topography, and parts of the
Okay,
kidney?
sorry.
What is the important-
Now, the interesting thing is, I can't remember if this was the video that had a lot of views.
of the renal cortex and medulla, and what's the sympathetic innervation of the kidneys? Hello, everyone. My name is Dr. Morton, and I'm a noted anatomist.
A noted anatomist. Anyway, so I'm going to go to some other place. I'm not going to listen to the video. You can listen to the video. Let's do the nephron. So I'm going to show you the nephron, and you guys can do this. And this is maybe how partly you can get images for your lab report. But remember, on Wednesday afternoons, I'm going to have people in this room. I showed you this image page. You can take pictures of stuff from his anatomy books. Okay, so the nephron is the functional unit of the kidney. It makes urine, and so like if you put that in a lab report or a test, the nephron is the functional unit of the kidney. How many times has that computer seen that? Tons. So you're going to kind of get dinged for that, and that's why I can't have 0% similarity score. But you should know it's like a snake-like structure, but a lot of times where it's shown in illustrations, sometimes it can be too simple of a drawing or almost too complex. So I'm going to go to images, and I'm going to find some of the most simple, and to show you how to interpret this. Okay, so here's a simple one. Notice how this looks like a snake. I'm over here, but that doesn't look like this one, and it doesn't look like this one. They're all anatomically correct, except this one is drawn like if you don't know the structure first. This is the one to learn on. So let me show you this. And we're going to end up with 3 important words that reflect what the nephron does. Okay, new tab, there, okay. So, this is a great picture for a beginner. There's some inaccuracies, but it's not bad. So the first thing you should note is the outer layer of any organ, not only the kidney, but any organ is called the cortex. So the cortex tends to be on the outer part of a structure. The inner part is called the medulla. So you have the medulla in the center, but the cortex, and it's hard to remember which is which, right? Cortex almost sounds like it's in the middle. And here's how I mnemonically always remember it. The C in cortex means, and this isn't anatomically correct, but it gets me to the outside. It's near the cover. Cortex is near the cover of the animal, of the organ, I should say. It's not the surface, but it's near. And then medulla is the middle. Medulla, middle, cortex, near the covering. Because there are other organs that have cortex and medulla. Does anybody know probably the most famous organ in the dog that has something cortex, something medulla? It's an organ that has a distinct cortex and medulla. Anybody? From a biology class? And see, here's what I don't know. I don't know what you have in your brain. I wish I did, because then if I knew everybody knew that, I wouldn't even have to ask it. How about the adrenal gland? Adrenal. You're going to find them in these animals. The interesting thing is, take away the AD of adrenal. What do you got? Renal. What's renal mean? Kidney. So there's a renal gland, often called a kidney, but then there's an adrenal gland. AD means near. So the adrenal gland is named because it's near the kidney. So you got renal glands and you have adrenal glands. And we'll show you that sometime. But the point is, sometimes the name tells you where something's at. So the adrenal gland has a very famous adrenal cortex. And the adrenal gland has a very famous medulla, the adrenal medulla, the adrenal cortex. And they put out all kinds of hormones. And they're near the kidney, AD, adrenal. So red often means blood, and it also often means arterial blood. So this structure is getting, this yellow structure, and I guess they put it yellow because it's making urine. This yellow structure, and we'll start over here, starts out by getting blood. But notice there's no red anyplace else. So blood comes into this structure and it leaves. So to improve this, I would put an arrow like showing flow. Not an arrow like this, because we'll have to talk about that. But an arrow bringing in blood and leaving with blood. But no blood here. So that's very important. So this is called the glomerulus, and we'll get another picture that spells that out. So this is the glomerulus, and the glomerulus is what's called a knot of capillaries, K-N-O-T. It's like a whole bunch of capillaries in this chamber. And that's strange because most capillaries aren't a knot. A capillary is this: artery, small artery, arterial, capillary bed, which is like strings, and then on the other side of a capillary bed, you have a venule, and then bigger veins. And what's the biggest vein in the horse? The biggest vein in the horse, vena cava. Okay? So what's interesting is there's something wrong with this picture. Let's say this is the incoming blood. It's coming here. That's called the afferent arterial. A-F-F-E-R-E-N-T. Afferent arterial. An arterial is the smallest artery. But it still means it's on the high pressure side. Do you know arteries are high pressure, veins are low pressure? So an afferent arterial has still relatively high pressure, but the drawing is wrong because this drawing makes the diameter of both vessels seem the same, right? That's not the case. If this is the afferent arterial, the diameter of this blood vessel is wider than this. What happens when you have a pipe that's bigger coming into a point and then a smaller pipe? Does that automatically set up pressure in here? Yes. So the amount of fluid flowing into the glomerulus is less than what comes out. And you might say, where does the fluid go? It goes right here. That's called Bowman's capsule. Fluid is flowing out of that knot of capillaries, but here's the kicker. No blood cells are leaving. The number of red blood cells, white blood cells, and platelets from last week's reading are going to be the same in here, but you've lost fluid. So, what's the packed cell volume of this vessel if I say this one's 45? What's the packed cell volume? Remember, 45 is a convention. There's 45% of the fluid of the whole volume is red blood cells. What do we got here? Friends and neighbors. 55. It goes up. Now 55 is a guess. 45% packed cell volume, but you lose fluid, so now in proportion, there's more red blood cells per unit volume. And that means packed cell volume goes up. If your dog is dehydrated, total body dehydration, the dog's packed cell volume is no longer 45. It's up, it's higher. Because dehydration means losing fluid, not losing red blood cells, white blood cells, or platelets. But it's mostly, and you notice how I'm saying always red blood cells? Because almost all the blood is red blood cells. The proportion of platelets and white blood cells is very small compared to the red blood cells. Oftentimes, there's 1,000 red blood cells to every 1 white blood cell. So sometimes I might say packed cell volume, it's all the cells, well technically it's just the red blood cells, but it doesn't matter because there's not much else there. So a dog that's dehydrated has a packed cell volume of higher than 45. So on a test, what if you ever saw the number 50 in that row of items? And it's talking about blood. Well, then you can say, gee, a dog that's hydrated could have a packed cell volume of 50 or higher. So you could use that from knowing what you did today and so forth. Okay, so afferent arterial means coming into a point, A-F-F-E-R-E-N-T, and then this is the efferent arterial, E-F-F-E-R-E-N-T, meaning outgoing from a point of reference. Afferent, efferent, used all over. So that's what I want to make sure that you don't sometimes think of a term might apply for just kidneys, right? Got to be careful about that. So then you get this fluid that's here, and where my arrow is, my cursor, that fluid would be called crude urine. Crude means not refined, and that volume of crude urine will never make it to the bladder. That's one thing about the kidney. Makes a lot of crude urine, but not a lot of refined urine. So we need to explain that. That's the physiology. If your kidneys made a lot of crude urine that ended up in the bladder, you're going to die of dehydration in a day's time. And you know, dehydration is a loss of body fluids, right? So then, the short story is, there's fluid here that's called crude urine, and then it travels this direction. And see, there's an arrow going this way. But if you had to know, remember how I put the arrow in there for blood flow? And then urine always goes this way. And then out. Sooner or later, this part goes to what's called the renal pelvis. And that's another thing. Pelvis, that's not just your pelvis, like Elvis shook his pelvis. No, where urine collects in this edge of the kidney, it's called the renal pelvis. That's connected to the ureters. So if pelvis is in a test question, it's unlikely that I want to hear about anybody's, shaking their pelvis, right? It's more like renal pelvis. So here's finally an arrow. This is called the collecting duct. Everything has a part. Every nephron takes relatively refined urine down to the renal pelvis through the ureters, what's the force moving urine from the kidney to the bladder? Peristalsis. Okay. And then, it goes to the bladder, and we'll talk about that later. Anyway, what are these arrows doing? Well, when you make crude urine, remember, no blood cells go through the little filter, but a lot of other things do. Like glucose. I'm going to pick on glucose. See, glucose is here. Why in the world do you want glucose to leave your body when you just ate a meal? You don't want glucose to leave. But it's in the crude urine. But I'll tell you right now, it's not in the refined urine unless you're diabetic and have so much sugar. So these arrows that go out, that means that molecule is being transported out of crude urine, not into space. Now this is where this drawing isn't good, but right here, there's a capillary bed. It's not drawn. It's not a glomerulus, but there's a capillary around this whole thing. So when anything leaves through the wall of the nephron, that's called reabsorption. And that re is very important. So this is the site of glucose reabsorption. But you can't say that's the site of glucose absorption. Where's the site of glucose absorption? The small intestine. So terminology is very important. When you eat a candy bar, whatever, right, there's glucose, and it gets absorbed into the blood. That's glucose absorption. That's why they've got what I call it reabsorption, because it was once put into the blood, now it's getting put back into the blood. Reabsorption. Very important term. Some textbooks might call it absorption, but it's technically not right, because glucose absorption is in the small intestines, right? The small intestine, its claim to fame is to absorb what's called monomers into the blood. Monomers means like one structure of something that gets into the blood in the small intestine. Monomer, M-O-N-O-M-E-R, a monomer. Things are broken down into basic parts that get absorbed. And that's the interesting thing. Look at how this is all connected. If your pancreas isn't working right, things aren't broken down into monomers. And the animal starves even when it's eating food. What's that condition called in dogs? When the dog eats a lot, but it's not absorbing the nutrients, and it looks like you're, it's your neighbor's dog, you might think they're not feeding the dog. This has happened to people where your dog has this condition and somebody will call the animal control people because they must not be feeding the dog. Look at it, skin and bones. And you can become skin and bones even if you're eating meals. Anybody know that condition in dogs? Anybody ever heard of it? Where the dog can't digest food? Okay, see how this all rolls together? It's EPI. And that stands for exocrine, that's E-X-O-C-R-I-N-E, pancreatic insufficiency. If you remember from a biology class, and see, I don't know if you've ever had this before, but the pancreas, one of the things it does, it puts enzymes in the lumen of the small intestine, and those enzymes break down everything into monomers. So you absorb glucose, but you don't absorb complex sugars, but they're broken down. So you can have a dog that doesn't put out enough pancreatic juice, it's called, that has the enzymes, and it can't break down the foodstuffs material, so they pass through, and the dog looks like you're starving it, but it's not. So did that ring a bell for anybody? Okay, so look at how many, I taught 90 people EPI, exocrine pancreatic insufficiency, and let me Google that and see if we can find a picture, because it's basically all related, right? I mean, you got to think of physiology and anatomy as all together. We're making crude urine, but we're saying we want that glucose reabsorbed. But I just told you a case of pathology, pathophysiology, where it couldn't get absorbed in the first place because the pancreas isn't working right. So let me do it. Okay, so now make sure you understand exocrine. Exocrine, see, look at it. See how it is? Exocrine pancreatic insufficiency, if you didn't know how to spell those words. It's a very common thing, happens in people. I think it happens in most mammals. Of course, what you should understand is most wild animals don't have this. And that brings up this whole discussion about why wild animals don't have something and your dog does. Does anybody know the short answer to that?
Do they not live long enough to get it?
That's partly right. They don't live long enough to get it, but there's a better answer.
Builds immunity?
Builds immunity? No. I mean, these are kind of, there's cases where all those answers are right. Here's what happens in the wild animal population. An animal that has exocrine pancreatic insufficiency, there's usually some genetic component to it. The animal will die and not pass on the genes. Only in animals that you fix things will things get passed on. So what's the most example? People, we fix things that happens in our kids, we want them to be alive. They might pass that trait on. Look at how many things are by the genes. You can do the gene thing and you can see if you've got the gene for cancer, breast cancer, that's something that's going on in women now. Okay, and dogs and cats, especially dogs, we fix things. We don't fix things in deer. We don't fix things in sheep. Right? You don't fix any of that. The animal dies and doesn't pass the genes on to the following generation, usually. So that's why all those animals don't have that. I mean, I always wonder, we live on the country, we have some acres, and I have trail cams up. It's amazing how many animals are out there. And you know what? Most of them look always pretty healthy. Why? Because over time, if some animal had genes for cancer, it died. It didn't pass the genes on. I mean, I got woodchucks, deer. I've got a doe that's got twins. I say I because I take, the camera takes pictures of it. I mean, and they are so healthy, and they're out there in the storm, and sometimes it doesn't rain, and I put some water out for them once. You know who played in the water? Raccoons. The whole family, 7 raccoons playing in the water that I left out for the deer. And they got it so dirty. I felt sorry for the deer. Anyway, exocrine pancreatic insufficiency only happens in humans, dogs, and cats. Why? The other animals die and don't pass it on. I should say it's rare. Okay? So let's do this just real quick. Because it is, so here's something you could write down. It's because the pancreas doesn't make enough digestive enzymes. Notice we're not saying anything about insulin or anything, hormone from the pancreas. Because the word exocrine means anything that's put into the digestive tract in a tube. That's what exocrine means. Something's going in a tube. So I call it pancreatic juice. Pancreatic juice is coming from the pancreas, flowing out a duct, D-U-C-T, and it's got the enzymes to digest proteins, carbohydrates, so forth. So then, let me do in dogs. See dogs up here? Yeah, I mean, because it's all over. I want to do an image thing. How come I'm not seeing that? Okay, so what I'm going to do is dog. And then in dogs. Okay, see, so there's no other, in this case, it's humans. Anything that doesn't have an animal with it usually refers to humans. Most of the stuff is humans. Dogs, okay, in dogs. And then I want an image. I don't know why it's not giving me that choice. I'm not going to spend a lot of time on it. Here's the one I remember. Look at that. If you saw this dog in a backyard, you're going to call the police. You should see him after treatment. I won't show you, but the picture after treatment is flabbergasting. He has EPI, and you might say they're not feeding him. No, and you should see how he recovered. I mean, it's like spectacular. Because, remember how, didn't I say that last week? If you're low of something, that's not bad because you could up. If you got too much, that's bad, right? Remember the hormone thing? If there's too much hormone, that's hard to battle. But if you have less hormone, then you give the animal. So in this case, you give the animal digestive enzymes in its meal. Spectacular. Good, I'm glad we got that image. Okay, so anyway, things leave the lumen, L-U-M-E-N, lumen of the nephron, and that's what these arrows show, and that's called reabsorption. Over here, when water left the glomerulus, that's called filtration, when you're passing something through a sieve. Blood's not going through, but fluid is. So you got filtration here, but also glucose is going through, sodium is going through, potassium, right? Vitamins went in the urine, you better get those back. Otherwise you're going to be deficient of those vitamins. Now here's something, let's see. Here's something that, oh yeah, here's one. Notice this arrow goes into it. It goes from a capillary, think of a capillary here, into it, into the crude urine. So that's our third process. One process was filtration, one process was reabsorption, and this process where things come from the blood and go into the urine, that's called secretion. And it's only because of those three processes do you get refined urine over here. So there are things that can be in the blood and that get transported into the lumen, and then oftentimes carried out in the refined urine to the bladder and then excreted, right? When you excrete urine, that's coming out of the bladder, right? So here's the kicker. If you have a kidney infection and you know the bad bugs are in the lumen of the nephron, that's hard to get to. What you do is you give the animal an antibiotic that's secreted into the lumen. So you can give an animal antibiotic, and it might get there too from the glomerulus, but it may be too large to pass through the filter. But here it's going to be, just think, you could write in here antibiotic, and some get secreted into the lumen. That's a great way to fight a urinary tract infection. That's called upper urinary tract infection. Upper means it's in the kidney. Lower is bladder and whatever. So that's the processes. That's a pretty good diagram. But then you'll see how others are not that clear. Like, look at this one. This is not a one to teach the first time. Sorry. See all that? But that's really the orientation of the structures. So I'm going to go through this and let's see what they've got, and I'll explain other things. Oh, look at that. Did I say afferent arterial? The other one didn't label it, but yes. And did they make it bigger? Did they make the afferent arterial bigger diameter than the efferent arterial? You'd have to say yes, right? Doesn't that look bigger than this? That automatically sets up pressure to expel fluid out of the glomerulus into the lumen of the nephron, which we'll call crude urine. Anyway, so there's blood vessels all around, and I think this diagram shows that. See this? All those blood vessels that are on the outside of the nephron. Remember how that first drawing has nothing on the outside? This is really the accuracy. So the nephron is completely surrounded by small blood vessels, arterioles and capillary beds. And that's for when things get reabsorbed, right? Glucose reabsorbed, all those things. Okay, so then, we won't belabor this point, but there's a drawing. Everything is three-dimensional in the real animal, but on a screen, it's got to be drawn, But here's the glomerulus, Bowman's capsule, right there if you don't remember that spelling. Then you've got, that's the efferent arterial, afferent. Look at, they've got that larger volume. And then they've got something called the proximal convoluted tubule. We'll do some of this in lab, but proximal is a term that means closer to a point of reference. And then the antonym of proximal is distal, further. And maybe the way to remember that is distal means further distance from the point of reference. You know, there's these little tricks you kind of need, you know. Proximal is closer to a point of reference, distal is further away. So then this proximal convoluted tubule is, convoluted means kind of like snake-like. That means it's closer to the glomerulus than the distal convoluted tubule, okay? So all these terms, neat, they apply to anatomy, but you gotta remember, you know, like again, pelvis, that's, you know, there's a renal pelvis, there's many uses. Question?
So in the proximal convoluted tubule, that's where like the crude urine and all the reabsorption.
Yeah, on that snake part, the snake, the first drawing I had, they're never like that, but it's a good way to learn. So the proximal is the part where the glucose was being reabsorbed. Distal is after that loop. It's called the loop of Henle, by the way. Yeah. And it's labeled that way because, see, here's distal. The reason it's labeled proximal and distal is because the proximal is the first part where crude urine will travel first. So it's proximal relative to the glomerulus. But the distal isn't far away from the glomerulus the way the true picture is, but it's called distal because the urine has to flow through the loop of Henle and get over to the distal. So like here, if this is the proximal, then you've got crude urine and it goes down, 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 and then it comes back up, and here's distal. Okay? So, I mean, and there's, every nephron has to have all that blood supply for reabsorption, for secretion. Very complicated structure, really. And they're microscopic. You don't see this with the naked eye. Nephrons are microscopic structures. Okay, but I mean, so the thing is, a person for their lab report can find some images that mean something for you, right? And not maybe anybody else. So then, let's see, let's go to ureters and the bladder. Because after urine leaves the pelvis, the renal pelvis, it goes into the ureter. And I think the last week we talked about there might be extra ureters. What was that called? What's it called when you have more than the regular number? Supernumerary ureters. They may or may not be abnormal, but you got to know about their existence. Because if you ever become a surgeon for a dog, cat, or people, sometimes people have extra parts. Some might be missing a kidney and they don't know it. That's the amazing thing about anatomy. Unless you do an examination of a dead animal after it dies, you don't know what it was normal or abnormal. And in the human population, there are people that say everybody that dies should have an autopsy. And in animals, it's called a necropsy, right? Autopsy is human, necropsy is animals. And you might say, why should everybody, in the human population? Because you get a better handle on diseases. Just think of the big picture. If you did an autopsy on every human that died, no matter how they died, you would learn more about the disease processes. Why? Because if somebody dies in a car accident, they might have had cancer. They might have had a tumor in the brain. But if you don't do an autopsy, then that person's listed cause of death, blunt force trauma, by the way, and then that's gone. But the person actually had cancer. If you do enough autopsies, you might see clusters. Have you ever heard of clusters of diseases in human populations? Clusters are where there's too many things are happening. Too many types of brain cancer. Never heard of that? Clusters? Yeah. So when you study diseases, you look at the distribution. Look at the cluster of brain tumors downwind from that chemical plant. And if you did enough autopsies, you'd get a better handle on things going on. And the person that died in a car accident might have died from a brain tumor 2 years from now. But you missed it because you didn't do an autopsy. So there's a lot of times, there's a good reason to examine a person, autopsy, animal, necropsy, to monitor things, to get a better handle on it. So anyway, we're going to look up ureters, because the reason I do this is to show you all the images. And you know, when I did this, and here's another nice thing, a lot of times, and this didn't occur when I was at your age or in college, the pronunciation of these words. We always had to learn it from our instructor, but I'll tell you what, I am surprised every year how many times past students email me. They're working for a pharmaceutical company or they're in a vet clinic, and they go, Rod, how do I find out about how to pronounce those words? You showed us in class, but I'm talking to people or I'm selling drugs, and I want to find out, you know, how to say these terms. And a lot of times now, like ureter, sometimes it's loud, and sometimes it doesn't work, but let me just click on it.
You were there.
Okay, I'll put it. Especially for some of the hormones, sometimes, you know, you need a little help. Because if you're like me, if I haven't said a word,
You were there.
I mean, I need help pronouncing it, basically.
You were there.
So the ureter is the tube. What's the type of muscle in this tube? Smooth muscle. What's the type of muscle in every tube in the body? Smooth. Everything that's a tube has smooth muscle. It's for contraction, and everything that has a tube usually has peristalsis. Think of that. Even the oviduct has peristalsis, moving eggs. I should show you when we get to reproduction. This guy, years ago, he was an MD, he made a movie of ovulation in a rabbit, I think. He had like this little camera, and the egg oozes out of the ovary. It's like it's getting paid by the hour. You know, if you get paid by the hour, you do things slower. The egg goes out of the ovary, and then cilia bring the egg over to the mouth of the oviduct. And I saw this when I was your age, and I've never forgotten it. And here's what happens when the oviduct senses the egg. You know, the egg is like a sphere. It's the size of a period at the end of the sentence. In the dog, cat, all of us, well, the women in here have eggs that are the size of a period at the end of the sentence. So this hand is the egg, okay? And then my other hand, I'm going to put the microphone down, but my other hand is the opening of the oviduct. Now the egg gets transported by cilia, little hair, to the oviduct. Watch what the oviduct does. It swallows it. Stunning. The oviduct swallows the egg. Now the egg is in the oviduct ready to get fertilized, but it wouldn't have got there if that oviduct wasn't perfect. If the oviduct had scarring on it, it may not have been able to transport the eggs. And in fact, if, for example, if a cow, because I got to talk about all animals, if cows have bilateral scarring of the oviducts, bilateral is one word that means both sides, BI. If there's bilateral scarring of the oviducts, the animal, cow, will come into heat every 21 days, but never become pregnant. The animal will be infertile. So even if an animal releases eggs, it's still infertile if the egg doesn't get fertilized. Because the oviducts are the ones that transport it down. But there are reasons, and you put that in your lab report. See, there's things that, you know, like when you do reproduction, say, gee, if something's both sides or something wrong, and we'll talk about when fluid collects into the oviducts, that's another case. If it's bilateral, then the animal is sterile, infertile, but will be in heat every 21 days. How many are familiar with cattle? 21 days, okay, there's a few. They have a 21-day estrus cycle, but they can be in heat every 21 days. You can breed them or have a bull. No chance of pregnancy if the oviducts do not transport the eggs to the middle of the oviduct. In the human population, sometimes this happens, it's called a ectopic pregnancy. I'll write that term down. Now these terms, make sure you don't connect them to a certain organ, because ectopic is a general term. You see, ectopic ureter, okay? So the word ectopic means not in the right place, not in the usual place. So look at what came up, ectopic ureter. It's a ureter that's someplace else. Then you've got ectopic ureter dog. Then you've got ectopic pregnancy. Now this would be humans. This is where a pregnancy starts in the oviduct. The egg was fertilized, but after fertilization, the egg is supposed to, now it's called zygote, right? Zygote. The zygote is supposed to go to the uterus and implant. But if it stays in the oviduct and it's not detected, it's going to wreck the oviduct. And that side's going to be sterile for the rest of the woman's life. We're talking about women now. But the other side is okay. So that woman can still have a chance of getting pregnant, but if she ovulates from the bad side, she's not going to get pregnant. And the ovaries never go like this, my turn, your turn, my turn, your turn. They never do that. Might be two on this side, you know, ovulation, then the next one on this side. Or it might be one back and forth once in a while, but there's no rhyme or reason. It's the egg that's going to ovulate. Anyway, ectopic pregnancy. That's when a pregnancy starts in an oviduct. And if it's one side, not a problem. You know, well, you're half the apparatus is gone. Anyway, ureter. So then, ectopic means more than the usual. Well, I'm going to say, let me get this up here first. Okay, so here's the normal system, right? This could be a dog, it's a human, but, you know, this is the aorta, the largest artery in the body, aorta. So the heart is up here. This is called cranial, this is called caudal, right? Cranial, caudal. This is the aorta. One blood vessel going into each kidney. And it doesn't show it, but then the blood leaves the kidney and goes into the renal vein. Renal artery, renal vein, aorta. Okay, and then you've got the renal pelvis, taking urine by peristalsis. And this drawing isn't correct. The ureters don't go in back there. The ureters always go in. See those two little dots? You see that? That's where the ureters connect. So, you know, they're kind of right, but it looks like they go in here. No, they go way down here. Okay, now the ectopic. So here's a kicker. You could label this ectopic ureter because it's a ureter that's not in its right location. Look where it's going. But you could also call it a supernumerary ureter. See that? So it's supernumerary ureter, and yes, it's ectopic. Because if there's an extra one, it never goes in one of those same openings. That's an opening to the bladder. So, okay, so that's ectopic, okay, and let's see, I'm not sure, let me see if I can find it on my desktop. Maybe I can't. Does anybody see where it says ureter? Look at my, here it is right here. Good. Thanks, I found it. I'm gonna show you how the ureter enters the bladder, and when you see it, you're going to understand why no matter how full the bladder is, fluid won't flow back up to the kidney. Because if it was just a pipe, if the bladder got too full, things would stop and maybe go up, and that's terrible. That brings bacteria out of the bladder up to the upper renal system, and you never want that. Everything should be going this way only. And when you see how it enters the bladder, the ureter, you'll go, oh, I can understand how if the bladder is full, I can still make urine. Look at the angle. Here's the ureter coming from the bladder. Look at how it enters from the kidney. Sorry. Oh, what happened? Oh, wait a minute. Okay, let's do that again. I moved something. So I'm going to be careful what I touch. So the kidney's over here, comes down the ureter, and notice how it goes into the bladder. This is the lumen of the bladder, and if the bladder got so full, wouldn't that press down this way? This flap would press that way. That's not going to stop urine from getting in, because remember, it's muscles that are moving the urine in, but it's going to stop urine from going up the ureter. What a nice little piece of architecture to help not have urinary tract infections. Do urinary tract infections happen in deer? Not really. Why? Because any animal that didn't make that connection that way died from a urinary tract infection, right? And the genes were stopped. So in all those animals, anything that has a genetic component is going to be gone. That's my story, and I'm sticking to it. See you guys in lab tomorrow. First one's 9:30. I'm available for questions up here if you need. I don't know why that went on to the next one. Yeah. Yes. Take care. What's that? Tomorrow, just listen like everybody else, since we've never done it before. Yeah, just come and absorb. Hi. It can be. Most things have a genetic component.
Okay, but like, is it like an addition of a gene?
I don't know the genetics, but here's the deal. You can't fix it. If the pancreas isn't making enough enzymes to put in the lumen to digest, in 99.9% of the cases, you can't do anything about it to fix the primary problem. You have to feed the animal an enzyme. Now, how big a genetic component it is, I'm not sure. And like, you get into the route of inheritance. Is it dominant? Is it recessive? I don't know. Because they really haven't done that many studies on it. Because it's hard. When you do studies, you need a bunch of animals that you keep in the same environment and do the same thing. Like, it's easy to do studies with cattle. Okay, I've got 40 cattle. I'm going to feed them this mineral, see if it makes a difference on milk production. There's not many studies done on dogs, except maybe like Purina does feeding studies. But there's not much done on pathological conditions. So maybe somebody out in the real world knows the genetic component, like if it's dominant, recessive, I don't know. I just know it's a terrible thing, that picture I showed you of that dog, but it's stunning how after he gets enzymes, you couldn't tell him from any other dog. I mean, it's stunning. He's a great anatomy dog. All the bones are there, but, it was just because of the enzyme. But can you see how